All right, I think people are still trickling on. I can see people joining, but maybe we'll go ahead and get started. It's my pleasure to introduce and invite Dave Spencer here to present on his work. He's been doing all kinds of work for years that's highly relevant and of interest to this group, applying various NGS technologies to assaying tumors in interesting and very applied and translational ways, and has a ton of background and experience that fits right in with this group. So I'm really excited to see this presentation and learn what's new. So why don't you take it away, Dave?
Yeah, sure. Yeah, thanks for inviting me. And again, thanks for rescheduling at the last minute last time. Yeah, so today I'll just kind of give an update on our efforts and implementation to use whole genome sequencing for clinical evaluation of mutations in primarily hematologic malignancies. And so, I mean, this is our approach is kind of packaged in an assay that we call Chromoseq. And I think that the approach, I mean, I say here, it's a platform for clinical genome sequencing of cancers. And I think it's really kind of a strategy that I think is useful and is really kind of the way forward for a lot of genetic testing. And other groups have used this approach in various ways, and I think it'll continue to be used in this way. Uh, and so let's just to start by kind of making the case for whole genome sequencing as a one size fits most strategy for comprehensive genomic profiling. And so this comes from the last several years, from the time that we started the Chromoseq project, really to today, and it's motivated by actual laboratory testing. And the fact of the matter is that labor is the most expensive component of most laboratory tests. And clinical DNA sequencing of any type really are among the most complex and therefore labor intensive tests in the hospital. I mean, I can't think of a more complicated one than, for example, targeted sequencing that has, zillions of steps, lots of reagents, many places to go wrong, and it takes a lot of time to do it, even if things are mechanized to the extent that you can do it. So it takes a lot of labor and makes them expensive. And when you think about it, whole genome sequencing is the fastest, technically simplest, and most scalable DNA sequencing method. You make a library and you put it on a sequencer, and you let the sequencing and the analysis do the rest. So I do think that if there's anything that whole genome sequencing has going for it, it's the simplicity, which kind of underlies our work in the area. And for some applications, and I think a lot of cancers, not all cancers, but many of them, whole genome sequencing is just kind of the most efficient and potentially, maybe not most accurate. That may not be the best word, but certainly most comprehensive and complete sequencing approach. So I think that these are really kind of the main points that I think will make whole genome sequencing win out in a lot of situations for cancer and other diseases as well. So we started this project studying acute myeloid leukemia, which is really, I think, one of the earliest examples where comprehensive genomic profiling makes sense. Using whole genome sequencing. And so here I'm showing, it may be very familiar to many of you, or to some of you. On the left is the table this is from 2017 of the genetic markers that are used for risk stratification in AML at that time. And actually, most of these go back earlier than that to 2010. Some were added in 2017, but most were there 15 years ago. And these are really important. If you look on the right, it's showing outcomes using these genetic markers across a large cohort of leukemia patients and showing that they are predictive of survival when patients are treated with chemotherapy. And so they are really kind of the cornerstone of risk stratification in this disease. And so this is where we started. because we knew a lot about this particular type of cancer, but because I think it really is one of the first cancers where there were so many different types of variants that you needed to know about to properly risk stratify. So with that in mind, we developed this assay. Uh, and we wanted it to be, and I'll kind of go over this again in a later slide, but we wanted it to be fast and tumor only. Tumor normal was certainly it's the best approach to profile a cancer genome, but it's complicated and it's more costly, and we didn't think we needed it for what we wanted to do. Um, and we also wanted to focus. We were sequencing the entire genome, but we really weren't interested in the entire genome. We were only interested in the types of variants and the genes and the targets that were clinically relevant. Uh, and we ultimately got this clinically validated and CMS approved for myeloid malignancy. So it's kind of just overview of what Chromoseq is. So again, the principles were at the beginning, and they still are, to be fast. We wanted to do things as quickly as possible and not take forever. That was certainly, we felt it a barrier, at least in people's minds at the time when we were developing the assay. Again, we wanted tumor-only analysis, which is achievable because we really kind of know what we're looking for the most part. We don't need the normal to serve as a control to identify the somatic mutations. The recurrent ones could be identified just based on functional classification. Focus on clinical relevant events. So what I mean by that is um, copy number alterations and copy neutral loss of heterozygosity that are large or that affect relevant genes. So at the time, we were trying to kind of emulate karyotyping, and so anything smaller than what you can see on a karyotype, we really weren't that interested in because it wasn't clinically relevant at the time. These lists has grown, of course, as time has gone on, and sequencing has become more commonplace. So the one thing I don't have here is that whole genome sequencing can scale to accommodate those new variants, new targets, etc. Uh, we also, AML is characterized by recurrent translocations or structural variants, and so we wanted to focus on those, particularly ones that we know are clinically relevant or that are novel but potentially functionally relevant. So we really narrowed down the list of reportable SVs quite severely to make it easier to interpret the test. And then limited small variant reporting, just genes that we know are relevant. So right now we're targeting 64 genes. That's probably even too many, but it's practical from an interpretation standpoint. And this entire sort of list of hotspots, as I call them, it really can be informed by tumor type. So we started with a smaller list because we were just focused on AML, expanded it a little bit for MDS, and as I'll talk about today, we've expanded a little bit further for other hematologic malignancies. And we really wanted to limit all these findings to a single page. And so this is an example of just a kind of this is a real case, a sample report where we were reporting all different types of variants on a single page with a nice little figure. And this really is kind of the essence of the assay. We sequence the entire genome, but the findings are just the ones that are clinically relevant. And so this is how we report the assay today. It's basically a single page. There are multiple pages, but the meat of it is the first page. Okay, so the lab approach, as I mentioned, whole genome sequencing is really the simplest thing to do in the lab. This is just a schematic. We use a bead-based tagmentation library prep. And it's offered by Illumina for blood and bone marrow. And it really takes just a couple hours to make the library. And then we put it on the sequencer, and we sequence on the NovaSeq X Plus now on 10B flow cells, and each run takes about 24 hours. So we can really, achieve, sample to final report in 2 days if the timing is just right. And this is our analysis approach. So, this is actually what we published initially. We used Illumina's Dragon to do the alignments because it's very fast, and then we had our own custom approach to do this targeted analysis, structural variants, copy number alterations, and gene mutations. And so we've since updated things. Dragon now performs essentially the same functions that we did ourselves. And so we essentially just use Dragon now for everything. We do have a custom sort of filtering reporting strategy just to kind of adhere to those Chromoseq principles. Um, but all the raw calls now come from Dragon, and we know it's still very fast. The entire workflow from Illumina Run Directory to report takes about 2 hours, and you do have to pay for Dragon, and so it ends up being costing about $25 per case to do it locally if you purchase a Dragon server. So the analysis workflow itself now we've updated it, and I'll talk about that in a second. We've implemented Nextflow, which is a really nice workflow language with a lot of really great features. It's fully automated. You can start the workflow, and then once the sequencer is completed, and then it generates summarized text reports that are really useful for somebody like me as a lab director to look at the output and so forth. And those feed into a reporting system that our pathology department here has developed. Um, and you can also, there's various other inputs that you can run the workflow on to analyze your data. So we run locally for the most part, but it is cloud capable. Nextflow makes that very easy, and Dragon servers are available on AWS. And it's not that expensive, that much more expensive to run on the cloud. And then again, the way we've constructed the workflow is the targets, the hotspots, and the filtering criteria is very sort of customizable. You can change the config files. There's some key files that apply these target-based filters. So those can be altered for any tumor type of interest. So I'm just going to go over this is old data, but just kind of very quickly, the initial performance from our paper. Establish the performance initially, we sequenced 263 patients, and we were compared primarily to karyotyping and then some FISH, and we were 100% sensitive using this approach for chromosomal rearrangements, gains and losses, defined by karyotypes, and 90% sensitive for gene mutations at a VAF of 5% or greater. So we were limited in the abundance of small variants because of the coverage that we targeted in this initial publication, which was about 50x. And we know there were essentially no false positives in this study, and everything worked. So a lot of the gains that we saw were because karyotyping and FISH were also often inconclusive or failed. Okay, I think this is a nice figure showing that we can get new information when we apply genomic technology over the conventional testing, which is not necessarily surprising, but this really just kind of brings it into focus. So this is a set of 68 patients. They were consecutively sequenced during the course of our study, and they're ordered chronologically. And every time there was a new finding, the blue line increases, and every time a new risk-defining finding was identified, a red line increases. So at the end of the study, there were 25% of patients had new findings, and 15% of patients had new findings that would have redefined their risk classification by the ELN criteria that I mentioned before. Okay, so that's kind of where we started. We published that paper. We validated it as an LDT in 2021. It was approved by CMS for reimbursement in 2022. And it's really since that time, it's been used for all new AML and MDS patients who come into the hospital as inpatients. And on the right is the kind of just the number of cases. So we started with 263, and then just increased over. It's been increasing over time because it's really part of the routine diagnostic workup now for these patients. After we got it CMS approved, because we need to get it paid for, and that's kind of important. So it wasn't really used as an outpatient setting where insurance has to pay the bill instead of the hospital. But now that it's approved, it really is part of the diagnostic algorithm for MDS patients in the outpatient setting, which is fantastic. And then along with that, it is part of two clinical trials, to just to further evaluate its clinical utility. And we've sequenced. This is a little old. Um, Now you can see it ended last year. I mean, it's really up to about 1,400 total cases whole genomes that we've sequenced. And many of those, say at the very end, many of those are consented under our banking protocol for research. So we can analyze those genomes and potentially identify new or kind of put more precision on genomic events in AML and MDS. Okay, so what I'll talk about from now on is really other cancers. So this I think this strategy of being fast, of being targeted, of kind of ignoring the rest of the genome and just focusing on the things that are clinically relevant is a good strategy, and I think it's the way forward. Sequencing is getting cheap enough that one can do that. We don't have to try to wring every last bit of data out of a genome. We can sequence it and look at the parts we need at that minute, and then kind of just put the rest away, or even throw it away eventually, and sequence it again if we need to. So is it useful for other cancers? And I think at this point, there's still a cancer type has to meet some criteria for it to be practical to do this, at least in our lab at our center. Uh, the first is whether mutational profiling is really used for clinically management. The oncologists in cancer are they gonna use this information? Because you have to rationalize the expense and everything, and so it can't just be for academic purposes, unfortunately. So that's the first question, and of course, the answer to that for many cancers is yes, and it's increasing, every year. So the next criteria is do clinically relevant mutations span multiple alteration types? So, by that I mean small variants, structural variants, copy number alterations, maybe even, tumor mutation burden or MSI, et cetera. If only one of these is relevant, if there's like a single, gene or a small number of genes with small variants, SNVs and indels that's really like 90% of the mutation spectrum in that cancer, then it really doesn't make any sense to do whole genome, honestly. So you have to, I think, but once you need multiple different mutation types, then it becomes a good option. And then again, at our center, is there a path to reimbursement? I mean, we can perhaps write a grant and so forth, but we have to be able to get paid to do it ultimately. Otherwise, it's not really useful to consider to expand Chromoseq to another tumor type. And then last, what specimen types can be obtained? So as I'll show at the end, we can do whole genome on formalin fixed blocks, DNA extracted from blocks. It works, but it's more expensive, and our rapid library prep isn't the best for that type of specimen type. It works, but it's not great. So that's certainly a consideration that we make right now. At some point we may switch our library prep type, and we could expand into solid tumors more easily. But right now, fresh tissue is an important criteria. Okay, so B-cell leukemias really, I think, meet these requirements. So what I'm showing here is a really pretty impressive UMAP clustering of about 2,000 pediatric ALL cases. This is just by bulk RNA-seq, showing just very nice clustering of these patients, and these clusters are defined by genomic events, and so they're labeled by they're colored different colors, and then they're labeled to indicate what sort of either gene mutation, gene fusion caused by a translocation or a structural variant, or copy number alteration. So in BLL, there's a lot of changes in just the chromosome copy number. And so we thought this was a great candidate for Chromoseq. So there are many different types of genomic events, many alteration types, lots of different genes involved that are difficult to capture using another approach. Not impossible, but challenging. The clinical samples are blood or marrow, and genomic testing is typically reimbursed. So we thought this was a great candidate for the next phase of Chromoseq. And so just kind of to drive home the point that these events are used for for clinical management. These are the international consensus classification consensus criteria for BLL listed here, and I'm just coloring whether the group is defined by a structural variant, multiple structural variants, copy number alterations, etc. Uh, so these are the established categories, and these are provisional categories here. So again, these are useful, and these categories are associated with differential clinical outcomes. So I mean, they're definitely useful. And so again, it kind of meets those criteria. And so one subtype I want to just I'll talk about in more detail, but is this BCR-ABL-like group. And so BCR-ABL-like was originally sort of identified using transcriptional signatures. So these patients kind of cluster with patients who have the BCR-ABL oncoprotein caused by 922 rearrangement, but they don't have that mutation. So they were called Philly-like. And it turns out that that this subgroup is really it's driven by a variety of structural variants. So here I'm showing some figures from this paper in 2017 that looked at adult patients with BLL, showing that about a third in this study, about a third of patients had the Philly-like subtype. Uh, and 90 other studies, not this one in particular, but have shown that up to 90% of the Philly-like BLLs have an alteration or an overexpression of a cytokine receptor or kinase gene. In this particular study, they found that 61% had alterations associated with a single gene, CRLF2, so which I'll talk about later. And then this is associated with poor risk. So again, it's clinically relevant, a clinically relevant subtype. So our goal was to validate Chromoseq for BLL. We thought it was a good application of Chromoseq, and it kind of fit all the criteria. So the goals of the project were to define the analytical performance, and to validate it in the clinical sequencing lab and use at our center. And then we were particularly interested in this Philly-like subtype, given the diversity of events that are associated with it, and the fact that testing um, for this subtype, it's complicated. And here we do flow cytometry for the CRLF2 surface marker to see if that's increased, and then we send it out for a variety of tests. It's kind of a hodgepodge. Not that it isn't effective, but it's complicated. And so we wanted to know how well Chromoseq could identify this subtype, because there were so many different rearrangements, and some of them are pretty complex. And so to do this, we made some updates, and I kind of mentioned it already, but we validated a new PCR-free library prep. We thought that would improve CNA calls, perhaps. And then we've increased the sequencing depth, so we're targeting 80x now instead of our initial study was around 50x, and so we're creeping up as sequencing costs drop. We try to sequence as deeply as we can. We added new genes to analyze for mutations that we know have mutations in BLL, and then likewise expanded our structural rearrangement analysis to include other genes that are part of this BLL sort of spectrum. And then our initial assay, we really didn't look for gains and losses, or CNVs that were less than a few million base pairs. But this is important in BLL as well as other cancers. So we've reduced that limit, and really look for any deletion or duplication that it could be clinically relevant, that affects a gene that we think is important. And then we added support for ploidy estimation, which is pretty complicated but important to identify whether there's a modal change in chromosome number. So the cohorts that we used for this validation, there came from two sources. So one was our internal. We had one grant. This is all grant funded, by the way. But the first was a cohort from WashU, just retrospectively banked cases that we picked to represent a range of cytogenetic changes we had cytogenetics in fish from clinical testing, and then we were able to perform RNA-seq on some of those patients. And so that's shown in the left column here. So total of 88, and we had RNA-seq on, about half of those patients, which we use for some orthogonal, confirmation of events. And then the other core is interesting. It's actually from a multi-center clinical trial run by the Alliance. And this was a trial of BCR-ABL negative BALL, where they randomized to receive a monoclonal antibody therapy plus chemo versus standard chemo. Uh, we had, this was part of a clinical trial, so things were more regular and not as selected. We had clinical cytogenetics in fish, and then interestingly, as an integral biomarker for this trial, testing was also performed using this 8-gene low-density array card that screened for a Philly-like expression signature. So, and this was an integral biomarker for the clinical trial. And then we also had a few cases at RNA-seq that were done by another, like a member of the trial for research studies. And so that's shown here in this middle column. So 136 total patients, and these were all young adult and adults, and showing the breakdown here. And so all of them had this Philly-like LDA card analysis. So we had a total of 224 cases to do our validation and kind of answer these objectives of our study. Okay, so first, just very simply, we had cytogenetics, and so we wanted to know the sensitivity for risk-defining events, and so we classified each patient by the ICC subtype, by Chromoseq and by cytogenetics, and then asked if Chromoseq missed any of these events. And so we only missed one event so we were nearly 100% sensitive for these events. Mostly rearrangements here. One case in the Alliance cohort very sort of vaguely observed an IGH rearrangement that they thought helped reflect an IGH CRLF2 rearrangement. This is one of the most common ones. It's unclear what we don't have much data on it yet at this point, but we did not identify this event, an event like that in the Chromoseq data. So that's a potentially false negative. But all the rest, we found an event that would put patients into these classes if it was reported by cytogenetics. But of course, we identified a lot of additional events, and so that's what I'll show in this slide. So just starting out here, using cytogenetics in fish, there were 119 total cases out of our 224 that were unclassified. They essentially were, BALL, NOS, no notable events by conventional testing. And then when you do whole genome sequencing, there are fewer of those. So we identified events in the difference here, and there are only 50 patients left over that were, essentially unclassified. And so these patients are classified into a variety of groups here, which I'm showing here. So red is Chromoseq and blue is cytogenetics. So large number of the Philly-like subtypes. There's actually two Philly-like subtypes depending on the rearrangement. So this one really dominated the group. We identified canonical class-defining rearrangements involving TCF3, PBX1, DUX4, MYC, etc., MF2D. So again, not surprising when you apply a higher resolution technology, and similar to what we observed in our initial publication of the assay. Now, as I mentioned, we had RNA-seq on a subset of these patients. Some we performed ourselves if it was our internal cohort, and in the Alliance cohort, we had a small number. And so out of 39 new findings by Chromoseq, where we were able to look at the RNA-seq data, 36 of the cases were confirmed by RNA-seq. And these other three, we don't have the raw data, so we're not really sure yet, but we're working on getting that to see, if this was truly a missed event or if there's signal of these events in the RNA-seq data. And so in green here is just to break down, we identified all of these events here were supported by RNA-seq, either with a gene fusion or overexpression of that gene. And then for 3 of the Philly-like JAK-STAT activated group, there was no support reported by the RNA-seq data from the Alliance trial. Okay, so now just focusing on the Philly-like BLL patients. And so again, this subtype is caused by a pretty broad range of SVs. And then we had this nice result from the Alliance trial from the 8-gene microarray, a small, low density array, which has been published before. It's been used, in a number of studies, and essentially the signal from these 8 genes is, undergoes they do a logistic transformation and to scale the signal, this aggregate signal from 0 to 1. And then for this trial, the protocol states that if the value is above zero point 0.1, then it's called Philly-like positive, or zero point 0.5 rather, it's called Philly-like positive. And if it's below 0.5, it's not Philly-like positive. So I mean, we have the raw data from this array, and so for the 136 patients in the trial, that the values are shown here, just sorted from smallest to largest, and then the classification that was given by the trial is indicated by the color. So red is Philly-like signature, a screen positive, and gray is negative. And so when you look at the cytogenetic results from the trial, which are indicated here, so each patient is a square. The red tiles are patients where there was an event that's consistent with the Philly-like subtype. So a fish positive result or something identified in the karyotype. If it's blue, then another ICC defining event was identified, and then if it's dark gray, it's unclassified. There's no notable event. So the few RNA-seq cases we have for this trial are shown here. Oh, and you can see that, clearly the red tiles sort of cluster at the far end here, which is, reassuring and, expected. The RNA-seq data I'm showing here, we only have RNA-seq data for a small fraction. And so, where there's no data, there's a light gray tile. But again, there's, clustering of the cases with the Philly-like subtype by RNA-seq at the right end of the plot where there's high signal by this screening assay. Okay, so then for Chromoseq, we had Chromoseq for all these cases, and you can see the same pattern. So, good concordance between, all three assays, generally, and the Philly-like findings cluster at this end of the spectrum of patients. And so, just a few things to note. So first, you can see there's kind of an inflection point in the scale in the LDA card value here, which maybe suggests that as you get close to the 0.5 cutoff, I mean, it gets a little bit ambiguous and maybe unclear which group the patient should really be in. So again, the second thing is the red tiles here cluster at the end where there's high values, which is good. And Chromoseq did, there are four patients where we didn't identify any event, but the LDA score is rather high, so those could potentially be false negatives. None of those have a conflicting finding by any of the other assays. Either cytogenetics didn't have anything either, or we don't have RNA-seq. So and we're ongoing work is to kind of adjudicate some of these discrepancies. And then finally, again, as you can see, as you get closer to this 0.5 cutoff, things get a little more equivocal. There are more cases where there's another definitive finding where you think would put the patient into a patient into a different category, or there was it's kind of no diagnostic finding necessarily at all. So in general, I mean, in general, this is kind of summarized here. So that the yield or positive predictive value of Philly-like findings, so structural variants or gene fusions in the LDA positive set. So Chromoseq is about 67%. RNA-seq, which is a much smaller number, was 76%, and then by cytogenetics was lower, as 22%. And then the negative predictive value. So, if we called something in a different in different category, did it have the Philly-like signature by the expression screen, and that's shown here. So most of the time we were concordant with all assays were concordant with the signature. Uh, we're still kind of, working on some of the kind of deeper findings from this cohort. So, I mean, one interesting thing is to look at the spectrum of structural variants in the Philly-like cohort. So these were really dominated by rearrangements involving CRLF2, which we saw in that prior study. So it was like 60% in that study had rearrangements involving CRLF2 or CRLF2 overexpression. And so here in this cohort, 81% of the Philly cases that we identified had CRLF2 structural variants. Most of them were involved the IGH locus. There's also a small deletion on chromosome X where CRLF2 is located that makes a fusion with an adjacent gene. So that was the next most common. And then there were rearrangements between CRLF2 and other genes. Some of these are complex rearrangements that may involve another gene and IDH that makes up another 7%. But by far, this is the most common event that we identified in this cohort, which is consistent with the literature, perhaps not to this extent. Um, and just to show, I think one of the benefits of whole genome sequencing is you can look over a broad range of breakpoints and identify events. And so this is what we do in Chromoseq. We scan a region that's over a megabase pair. It's actually larger than this. This is the observed distance here in this study, but we scan over quite a large region at the IGH locus looking for these breakpoints. And so what's shown here it's a little hard to see probably, but the blue dashes in this track show the locations of IGH CRLF2 rearrangements from this study. And so most of them are clustered in this one small region, but you have breakpoints that go across the entire extent of this region. And so we're able to scan reasonably large regions, looking very specifically for these events. And likewise, the CRLF2 region itself, I mean, nearly all of the breakpoints are, they're not in the gene, they're upstream. So IGH, the rearrangement places IGH upstream of this gene, drives its expression, and that breakpoint can be quite far away from the actual promoter of CRLF2. So, about 70 kb away. So, and our strategy is for specific genes where we know or have commonly rearranged with a promoter with a strong transcriptional activator like IGH. We kind of bracket a region upstream of the gene so that we can identify those and call those to the top of the report and make sure that they're reviewed and reported. Okay, another complex rearrange locus that we're able to identify is DUX4. So DUX4 is highly repetitive. It's at the end of chromosome 4, and there was an approach using whole genome sequencing that was described in this paper a few years ago. It's a custom analysis of reads at this region essentially to identify rearrangements. And so this is actually now implemented in the Dragon software. And so we beta tested it initially, and now it's available just in the regular release. And we identified in our initial analysis, just 4 events that had these rearrangements, and we had RNA-seq for those rearrangements, and we can show pretty convincingly that expression of DUX4 is dramatically increased in cases with that rearrangement. So that's shown here. This is from our own internal cohort. We actually have 47 cases with RNA-seq, and then these red bars are the cases with this DUX4 rearrangement, and the expression in here. This is shown in TPM is just is super high. All the other cases have you can't even see the bars here because they had 0 expression in the other cases. So to support that these events, are these rearrangements are functional and IgH is activating DUX4. And we have similar support for some of the other IgH associated rearrangements, like CRF2 is another example where we can just we can confirm that with the expression in the RNA-seq data. And because we're sequencing the whole genome, we can obviously target, essentially, reasonable, arbitrary but reasonable number of genes for other events. And so we look for, for smaller deletions and duplications in genes that we think could be relevant. And then we can look for gene mutations. And so this is just showing CNVs and gene mutations that co-occur with the Philly-like subtype as we identified it, and it really a striking number of events affecting CRLF2, IKZF1, PAX5, which is known. People have reported this, but we're able to kind of really enumerate them and catalog them here. So these are deletions in IKZF1, PAX5 are extremely common, and then actual gene mutations. And over 30% of the patients with a CRLF2 rearrangement also have a gene mutation, which is interesting. And so we're able to capture all that this assay. And so some of this work we're still kind of going through the co-occurring mutations and some other aspects that really weren't part of our clinical validation, but are certainly interesting in this cohort. Okay, so the current status of Chromoseq now at WashU for heme malignancies is, we have completed the validation in the lab as an LDT for AML-MDS, which was already done, and both B and T ALLs, and then also myeloma. So that part's completed. We're still waiting for some aspects of kind of the bureaucracy to really roll it out for routine testing. ALLs really aren't that common, so we don't expect too many. Although I think we've already had two cases that we've sequenced, just even though we're not like fully sort of advertising it. And then myeloma is one we're poised to really fully roll out, and we haven't started that yet because there's kind of a different process needed for testing myeloma patients. And then the next step really is to go kind of the route that we went previously. So we need to get this, covered, and we can get it paid for in our local hospital, because it's paid just straight by the hospital. But if we wanted to get paid by insurance and so forth, we have to submit a new application to expand the indication to these tumor types. And we've been doing the assay now for several years. We run it 3 times a week. Turnaround time is 5 to 7 days, and it's really part of standard of care. And the oncologists are used to it, and they're really finding the data valuable. And that's one of the clinical trials that we have underway. We're simply just kind of keeping track of all the findings and the comparison with cytogenetics and what the oncologists who use the assays, how they incorporate it into their management of the patients. Okay, so that's the current status. So ongoing and future work. So again, the BLL study, I mean, there's a lot of interesting findings there that, many of them are been reported, but I think we can add to our, growing knowledge of the genomic landscape of BLL just with using our approach. We actually are able to do long read sequencing on about 100 of these cases. And so I think that's going to be really fascinating. Obviously, the samples that seem to be discordant is a great first sort of pass analysis of the long read data to see, did we really miss something, or is there some novel finding that we didn't recognize that's there. There's also other interesting aspects of long read sequencing like DNA methylation that my lab is heavily involved in. So we're gonna use these data to investigate that as well. And then for our study, the clinical trial has outcomes. And so something I didn't show here, it's not quite ready yet, but the simple question of how to define Philly-like ALL, for example, and what the outcomes are, depending on how you classify it, I think is really interesting. And so we have the data. For that, we have to just coordinate with the clinical trial consortium to do that analysis. And so I think that'll be great. And then just other things going on. We have about 1,000 consented AML and MDS patients that we've sequenced. And so I've recently just completed just reprocessing all those cases kind of with a uniform improved approach. And we think that this will be really interesting to publish and publish the kind of the spectrum of mutations, both, just things like the breakpoints of common SVs, but then also maybe rare but recurrent events that are known about, but maybe they have sort of less information in the literature. And then analysis for recurrent non-coding mutations, and we've already found several in this data set that are interesting. And so that work is going on, and really kind of, again, it's kind of the more academic sort of research benefit of doing this assay. But I think it'll be informative for just again kind of adding to the landscape of the genomic events in these types of cancers. And then so more research efforts are going on for other cancer types. So we have sequenced several solid tumors, and then other folks are interested in applying it to lymphomas. And so for the solid tumors, we've actually been working on it for quite some time, and we're just about getting ready to kind of wrap it up a little bit. This is just a brief example of, just a one table and a figure from our study so far. So we sequenced 52 total solid tumors, a variety of types that we thought could benefit from this type of testing in one way or another, although it is dominated by lung cancer. And this kind of just summarizes the results here. And so the green is kind of in a nutshell. So out of 25 events that were reported by like FISH or other technologies, sequencing using the Chromoseq approach to detect 18 of them. So not great sensitivity. There were new fusions detected, either in cases that had prior testing, but did not report them, did not find them, or cases that didn't have any testing at all. And so really the main limitation in this study particularly is just how the quality of the whole genome sequence data. I mentioned that FFP DNA it is more fragmented, and so essentially your yield in unique genomic information per read sequenced is less, so you have to sequence more. And on our current library preparation type, it just isn't really that great for it. So we think that technically we can overcome some of these, with just by changing our process. And so we're kind of working on that now to see if we can improve the sensitivity in using FFPE derived DNA. But it is possible, certainly. Okay, so that's it. So I just want to thank a few people. So Division of Oncology, these are the oncologists that use the testing that we provide. So we're really grateful for that. And Jeff McGee in pediatrics, who gave us some of the pediatric BLL samples, and then the Department of Pathology. They're the folks who sign out the tests and do a lot of this work. Once we finish the technical part in the lab and the analysis, so we're grateful to them. And then on the right are the Shelley, Feiyu, Andy, and Tracy in the CLIA lab. I mean, they manage that lab and keep it running smoothly, and then the lab staff do excellent work day in, day out to do the testing. So I'm really grateful to them, and then our funding sources. So I think that's it. I'm happy to answer any questions that anybody might have.
Awesome. Thanks, Dave. That was great. Um, I, you showed an example report early on. Do you render a like risk stratification judgment, or you just sort of say, "Here's what we found. I didn't notice."
Yeah, so that was probably a really early version where we don't do that, but yes. So for AML, certainly. For MDS, it's a little complicated because some of the holistic classification kind of depends on hematologic parameters at diagnosis, and so some patients come in after they've been treated or whatever. So, and the most common the oncologists who really are users of the test for MDS, they like to do it themselves. So I don't know. Yeah, I don't know whether that for MDS is rendered for every report. ALL, yes. So an ICC classification will be generated. Again, we haven't done too many of those. So, but that's the plan.
Makes sense. Do you have access to everything you would need in order to do MDS, or you just don't even have the clinical information?
So, I mean, they have everybody, the sign up folks have access to it.
Mhm.
I mean, I just know there's some debate about the time, which peripheral blood sample they should read the hemoglobin from and so forth. And so there's definitely access to it.
Got it.
Pathologists, they can load up Epic or Copath or whatever here and get that information. Just my understanding is, yeah, like Megan Jacoby and Matt Walter are some of the, main MDS people. And, I mean, they like to do it themselves and they get irritated when it's incorrect for some reason that, is kind of opaque to the person who signed out the test. So, I
You
mean,
got another
it's not
customer.
like they're fighting. It's just, I don't know.
Yeah.
Megan doesn't want us to do it. We won't do it. That's fine.
Yep. Understood.
So,
Thanks.
Any other questions from the group?
I have one question, maybe two. Yeah,
Right.
thank you very much. Nice presentation. I think two questions. One question is because of the whole genome sequencing approach, once you identify structural rearrangement, it sounds like you follow up with RNA sequencing to identify like whether they lead to the like fusion. Do you follow up with RNA sequencing?
no.
Oh,
This
okay.
was just so the RNA-seq here was, I mean, to validate the assay, we needed some orthogonal data to support
Okay.
new findings. Yeah, it's not part of our routine sort of testing process. That was just for this validation study. Uh, and so actually, interestingly, there's one company that's starting to do Chromoseq now, and they're very big and they have lots of money and whatnot, and they've been doing paired RNA-seq on all their validation cases. And I talked to the medical director at ASH recently, and I mean, he says that they're still going to do RNA-seq, but they've never found a discrepancy in AML and MDS anyway. So, so I think that's good news. I mean, we find that our approach is pretty accurate, but it's always nice to hear other people say it, especially compared to RNA-seq, which we really have kind of outside of this study I showed today. I mean, we haven't done as much comparison between findings from Chromoseq and RNA-seq.
They never
Oh,
found a discrepancy in the sense that they never failed to validate something that was found by Chromoseq, or is it
Yeah, and
bidirectional, like everything that was nominated by RNA-seq that someone thought was believable was also found in the Chromoseq? Or
I mean, I haven't seen the data, but that's what he implied. Yes.
because that was one of my questions was like, is there anything that is detectable in the transcriptome that's hard to detect with the WGS assay for various reasons?
I mean, so there are just a handful of discrepancies, some of which I pointed out here, and the discrepancies that we do have. So in the data that I have that I generated myself, I didn't find any discrepancies, but numbers are small. In some of the data from that we got from an external lab where we don't have the actual raw data, there are discrepancies, and I don't know why those are. Those are generally events that we found by Chromoseq that were not reported by RNA-seq, and so we need to figure out like what's going on there and ideally get their raw data and analyze it. So I think that the I mean, it does seem like the concordance is very good. I think that there are regions that are complicated like DUX4, and so I would anticipate like that would be an excellent candidate for a discrepancy just because it is extremely complicated and repetitive. And so the customized method that's used to identify those events seems to work fairly well. But if it's a non-standard partner or something, then I would guess that I mean, yeah, I would predict that the sequencing the DNA is going to be less sensitive.
And I guess eventually we might have, I don't know if there are any examples right now, but like clinically interpretable things that like trans-splicing or circular RNAs or something that are,
Right.
yeah.
Right. Yeah, at this point, yeah, you're right. I mean, if there's a biomarker that isn't, is opaque in the DNA, then yeah, you're going to need something else for sure, for sure. At this point, those are relatively rare, at least in the
Okay,
cancers that I'm
another
thinking of.
follow-up question. Then for this complex structure rearrangement, how do you report like clinically versus how do you, I guess keep a record for your own clinical lab, just tracking the recurrent events? Like, do you really report the chromosome like coordinates, or that's way overcomplicated for clinical side to consume? You rather just say here's like hotspot, between the gene A and gene B.
So we report so complex events. I'll just give you an example. So one of the cases in the cohort that I presented, there's IGH is rearranged with chromosome 8, and chromosome 8 is rearranged with chromosome 9, and chromosome 9 is rearranged with CRLF2. And all of those events are identified, they're in the report, but none of them are identified as being, like a hotspot because the pair, the rearrangement pairs are not ones we expect. So like IGH and CRLF2. But when you look at all those events are identified, and so it's a very tricky one, but it's up to like people like Drew Hughes to sign them out to say, wait a minute, like there's definitely an IGH rearrangement with this locus, and then this locus rearranges with another gene that I'm familiar with. So then he will report those if they pass his sort of review. And then I mean, how do we keep track of that? I mean, we keep track of every variant that is in the report, and those are in the report. So we have we have the coordinates, we have everything about those variants. That particular example, I mean, it's hard to like add that to the hotspot. We may never see that event again, for example, but we do have record of it, and it does appear in the report when people are reviewing the case, and they can make a call whether they think this is a legitimate event. Does that answer your question?
Yeah, thank you very much.
Yeah.
I had additional questions, but I see we're out of time, and I know people have their next meetings to get to, so maybe we'll leave it there, and I will ask my additional questions some other time, Dave.
Okay.
Bump into each other in the hallways or something.
Sounds good.
All right. Yeah, thanks
All
so much.
right.
That was really awesome.
Yeah, thanks, guys. I
Yeah.
appreciate
Very,
it. Take care. Have a good holiday.
yeah, you too.
Yeah.